now I'm going to talk with you about your labs and what I expect for you to turn in for those lab assignments each week. Um, but before I do that, I forgot to mention I didn't go through the syllabus, and I know you can read it, but I do want you to understand how much your assignments, your graded assignments count. So the way that the grades are done in this class is your homework and your quizzes will be 5% of your grade each. Um, your labs will count 30% of your grade, and your midterm exam and your final exam will be averaged together for 60% of your grade. So the midterm will count 30% and the final will count 30%. And you will study for those by studying the homework and the quiz. The homeworks and the quizzes are where the questions come from. So just wanted to go over that very quickly with you, but I really want to focus on labs. So let's look at the very first lab that you have to do. It's a virtual lab, so no photographs are needed or necessary for this lab. This is the assignment where you turn the lab in, and it says the instructions and lab report document are located above this assignment. Once you have completed the lab report, including tables, graphs, and questions, upload it to this assignment. You're going to click on this. It's a Word document with some links. There's some links in the Word document. Okay, enable editing for sure. And then I actually sometimes have students do this lab really, like not virtually, but because this is such a quick class, that you're just going to do a virtual lab for what makes a cricket chirp. So some say if you listen to the sound of a cricket chirping, you can determine the temperature. Is it true or is it just an urban legend? Do any other factors affect how fast a cricket will chirp, such as humidity, wind, atmospheric pressure, or nearby crickets? So you're going to use a virtual lab to test a cricket subject. And then this is your link. I'm not able to just click on it, so I'm going to copy and paste it. Okay. And I'll just minimize that for a minute. So this will require Adobe Flash Player, which is available until January 1st, and then it won't be anymore. So I'm glad we have it because I like this lab, but this will be the end of being able to do this lab for any of my future students. So okay, so there's a tutorial. I will not go through every single thing, but there's a tutorial that goes over the scientific method with you. You should do every single part. You should click on the tutorial, go through every single part, and then click on the cricket icon and begin the cricket experiment. Okay, there's a lot of information that you need to go through on your own. I couldn't hear that chirp, so I wanted to see why. Okay, for some reason I couldn't hear it, but it says that, for example, the date is March 21st, the time is 6 a.m., and the chirping is slow. Several months later, you hear him chirp faster. Chirp, 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 chirp. And then So this is making you curious, basically. So you want to find out what's making them chirp, slow, fast, medium, whatever. What's determining the rate of the cricket chirp. So which would be a good statement of the problem? Crickets seem to chirp at different rates. Is there a single factor that affects the chirping rate in crickets? I wonder why and how a cricket makes his chirping sound. Well, we're not going to be able to answer C. We already know that crickets seem to chirp at different rates. And what we want to know is we want to discover, we want to study that and see if there is a single factor that affects the chirping rate in crickets. And then you're going to collect information. You'll read this information. I don't want to go through it because you can read it. And you're going to formulate a hypothesis. And I'm just going to click one. Should atmospheric pressure, humidity, number of crickets nearby, and wind speed be varied in this experiment? Since we are just going to experiment with air temperature on this particular one, then no, the other factors should be kept the same, right? That's how you do a controlled experiment. So this is letting you know the factors, the other factors, they're going to be the same for every cricket that you test. Now you're going to perform the experiment. So we want to do between 5 and 45. So let's go ahead and just do 5. And it counts. I don't know why. I wish you could hear the crickets chirp. But anyway, it might be annoying. The average number of chirps per minute is 4 over 10 runs, and we have it at 5. So let's go back to our document. And the document also gives you the instructions that you need. And every time that there is a question to answer, like for example here, put your hypothesis in red. Make sure that you write it, whatever it is. You don't have to do it in all caps, but make sure it's in red because that helps me to keep the answers distinct from the questions. Then the independent variables in the 5 different experiments. The one we're doing right now, for example, the independent variable, just so you know, is the one that you change in an experiment. So the one we're doing right now is temperature. Remember to do this in red. But you will actually vary some other conditions throughout the experiment. Sorry. Okay, identify the dependent variables. So that's always what you are measuring, and that's going to be the number of chirps or the rate of chirping. Record your data in the tables. Why is it important to test only one variable at a time? I'm going to let you think about that and answer that one on your own. But this is wind speed, so let's go down to temperature. So the temperature in Celsius is 5. The chirp rate was 4 chirps. I think it was 4 chirps per second. Yeah. And the controlled conditions was everything else. What was it? Pressure, humidity, all those conditions that we controlled. So you'll put those in there. When you get done, you will save this file and make sure you know exactly how you saved it. I'm not going to save it, but you will. And then after you save the file, you will go back to the assignment and you will click on the assignment and it will allow you to upload and submit that file. You will need to click on submit to make sure that it goes through. Now, these little video clips that I do only give me 15 minutes to do them, but I think I have time that I can show you how to do a kitchen lab as well. So the lab report for the acid-base lab is here. You will have to turn that in, but you will also have to turn in some photographs. With every lab that you turn in to me that you do that is hands-on, you must take photographs of yourself. You must be in the photographs, and your experimental setup before and after. I must be able to have, I need proof that you did the lab, not that you got the information online, or, I mean, you can even find pictures online of these labs. So I need you to be in the picture, and you can attach those pictures, those photographs, to the lab report. You can just scroll down to the end of the lab report and attach those photographs. Um, so in this particular lab, you are going to make an indicator. An indicator is a chemical that will change colors in certain conditions. So it says red cabbage, but when you buy the cabbage, it's not red, it's purple. So you're going to take some red cabbage, purple cabbage, break it up a little bit, put it in some water in a pot on your stove, and you are going to boil it. After it boils, you are going to turn the heat off and remove it from the stove. I mean, you're going to put it on a surface that is not hot. So, and let it cool, okay, because you don't want to get burned. So what this cabbage juice is going to do is it's going to change colors at different pHs. And you have a color chart here to help you see what the colors should look like. It will go from very, very acidic pH of 0 is going to be bright red, and very, very basic is going to be bright yellow, and all the other pH values will be different colors in between there. 7 and 8 are 7 is neutral. So a shade of purple is going to be neutral pH. And then this just tells you that if it's pink, it's an acid. If it's dark red, like a deep brick red, it's an acid at pH 3 to 4. If it's violet, any shade of violet or purple, then it's anywhere from a weak acid at pH of 5 and 6 to neutral pH 7. But as you start to see blue in the color, it's becoming a base. Blue is a very weak base at pH 8. When it starts to turn green, bluish green and green, that's pH 9 and 10. And then very strong base, green, the green yellow color is going to be pH 11 and 12. And so what you're going to do is you are going to test 10 liquids. And what you're going to do is, let's see. You are going to write the substance down in the box, and it's really anything that you choose. I'm just going to put tap water here. I want to know what the pH of my tap water is. You definitely want to pick some things that you already know are acids and bases. So I'm going to do white vinegar. And I'm going to do something that I know is a base is going to be just like a teaspoonful of baking soda, or half a teaspoonful in a cup of water, and that water can be tap water, it's fine. But it's going to be a baking soda solution. And that's going to be, those are things that we know. Vinegar is acidic, and we know the baking soda is going to be basic. Then you can pick any other 7 chemicals that you would like that are in your home. There's some they just need to be clear. The color needs to be clear. If you're going to use a soda, use Sprite or 7-Up. You know, if your solution already has a color, it's going to skew your results, since we're looking for a color change. And it says, make sure you read the very bottom of this assignment and turn in all the components, including the photograph, for full credit. And I'm going to do an actual demonstration of this lab, so I'm not going to keep going through it. But the main thing is you will not get full credit if you do not turn in a photo. Just make sure that you turn in a photograph of yourself and the experimental setup before and after you've completed the experiment.